Hello everyone, welcome to my channel Cells and Genes. So today I'm going to discuss on the topic ELISA. What is ELISA? ELISA is enzyme linked immunosorbent assay. It is an immunological assay which is used for the detection of a specific antigen or antibody. They are generally carried out in 96 well plates. Detection is carried out by the addition of a substrate that may generate a color. The commonly used enzyme labels are alkaline phosphatase or horseradish peroxidase. So here mainly I'll discuss two types of ELISA. They are indirect ELISA and sandwich ELISA. So let us discuss indirect ELISA. What are the steps involved? Antibody can be quantitatively determined by indirect ELISA. Here, the antigen is coated on the microtiter well. A serum sample containing the primary antibody is added to the microtiter well and allowed to react with the antigens. So this is seen in the second picture. The free antibody is washed away and bound antibody to the antigen is detected by adding an enzyme conjugated secondary antibody that binds to the primary antibody. So this is seen in the second picture. The unbound secondary antibody is washed away. A specific substrate for the enzyme is then added. The enzyme hydrolyzes the substrate to form colored products. The amount of colored end product is measured by spectrophotometer plate readers that can measure the absorbance of all well of 96 well plate. Now talking about sandwich ELISA. So we can see what are the steps involved in this type of ELISA. The antibody is immobilized on a microtiter plate. Sample containing unknown amount of antigen is allowed to react with the immobilized antibody. The well is then washed. The enzyme conjugated secondary antibody, which is specific for the different epitope on the antigen, is added and allowed to react with the bound antigen. Okay, so this is enzyme conjugated secondary antibody, and it is allowed to react on a different epitope on the antigen. Okay, any free secondary antibody is removed by washing. In the next and the final step, the substrate which is specific for this enzyme is added and the colored reaction product is measured using a spectrophotometer. Now let us look into the advantages. The first one is easy to perform with simple procedure. The second one is high specificity and sensitivity, and then high efficiency. Then it can be it's like no complicated sample treatment. That means simultaneous analysis can be performed without complicated sample pre-treatment. It does not involve any radioactive substances or any large amount of organic solvents. It is just antigen-antibody reactions. And it is generally safe and eco-friendly. Now let us talk about the disadvantages of ELISA. So it's labor intensive and expensive. So to prepare antibody, it is labor intensive and expensive. This is because it requires like sophisticated technology and also experts, because we need to prepare cell culture medias, we need spectrophotometer and all those things. So that it says like it's labor intensive and expensive. The second one is false positive and negative results, because there is a high possibility of false positive and negative. This is how is like if there is insufficient blocking of antigen, it could lead to false results. Next is like storage and transport. Refrigerated storage and transport is required, as we know that antibody is like a protein, and the antibody is unstable, so it requires proper storage and transportation. That's like refrigerated storage. So this was a short and quick video on ELISA. Hope you all understood it. So if you have any doubts, please do comment below so I could answer it. So once again, thank you for all watching my previous videos and commenting the doubts. So please subscribe to my channel Cells and Genes if you have not done yet. Thanks for watching. Thank you.